Welcome, dear students. Today we are going to discuss an important topic on structure and secretions of pituitary gland. The main objectives of today's lecture are to unravel the anatomical structure of the pituitary gland, to understand its physiological relevance, to study the hormonal secretions of anterior lobe, and finally we will conclude with the hormonal secretions of the posterior lobe. So let us start with pituitary gland. The pituitary gland usually has been thought of as the master gland because its hormonal secretions control the growth and activity of three other endocrine glands that include the thyroid, the adrenals, and the gonads. Because the secretory activity of the master gland is itself controlled by the hormones that originate in the hypothalamus, it is perhaps better to think of the pituitary gland as the relay system. The pituitary hormones are not limited in their activity to regulate the endocrine glands. They also act directly on non-endocrine tissues. Secretion of all these hormones is under the control of signals arising in both the brain and the information collected from the environment. Now, let us have a look on the anatomy of the pituitary gland. The pituitary gland is a small red-grey gland about the size of a pea, weighing roughly about half a gram. It is located in a holosaphenide bone that hangs from the base of the brain by a short infundibular stalk just below the hypothalamus. The pituitary gland is divided in two lobes with distinct embryological origins. Now, let us have a look on the structure and function. The anterior pituitary lobe is also known as adenohypophysis, while as the posterior lobe is alternatively called as neurohypophysis. That is connected by the median eminence of the hypothalamus by the pituitary stalk, also known as infundibulum. In some vertebrate species, for example fish, reptiles and amphibians, a distinct intermediate lobe, also called as pars intermedia, is present as a part of the adenohypophysis, where they secrete melanocyte stimulating hormone that plays an important role in controlling the skin pigmentation. Now that we have got a glimpse about the basic structure of the pituitary gland as shown in the figure, let us now proceed with its blood supply. The anterior lobe is sparsely innervated and lacks any secretomotor nerves. The anterior lobe is linked to the brainstem by the hypothalamo-hypophyseal portal system, through which it receives most of its blood supply. The superior hypophyseal arteries deliver blood to an intricate network of capillaries, the primary plexus, in the median eminence of the hypothalamus. Capillaries of the primary plexus converge to form a long hypophyseal portal vessels which course down the infundibular stalk to deliver the blood supply of capillary sinusoids interspersed among the secretory cells of the anterior lobe. The inferior hypophyseal arteries supply a similar capillary plexus in the lower portion of the infundibular stem. These capillaries drain in the short portal vessels which supply a second sinusoidal capillary network within the anterior lobe. Nearly all the blood that reaches the anterior lobe is carried in the long and short portal vessels. The anterior lobe receives only a small portion of its blood supply directly from the paired trabecular arteries which branch off the superior hypophyseal arteries. In contrast, the circulation in the posterior pituitary is unremarkable. It is supplied with blood by the inferior hypophyseal arteries. Venous blood drains from both lobes through a number of short veins into the nearby cavernous sinuses. The portal arrangement of blood flow is important because blood that supplies the excretory cells of the anterior lobe first drains the hypothalamus. Portal blood can thus pick up chemical signals by neurons of the central nervous system and deliver them to the excretory cells of the anterior pituitary. Now that you have understood the importance of portal blood supply in anterior lobe, let us directly move ahead and talk about its hormonal secretions. The anterior pituitary secretes 6 principal peptides, namely follicular stimulating hormone, luteinizing hormone, adrenocorticotropic hormone, thyroid stimulating hormone, prolactin and growth hormone. You can remember them by a simple mnemonics, that is flat PG. All these peptide hormones secreted from anterior lobe are better known as tropic hormones, with exception of growth hormone, that is GH, and prolactin. All have their major effects restricted to specific target organs. The posterior lobe secretes ADH and oxytocin. Now that you are aware with the names of the hormones, let us understand their physiological functions. The anterior lobe secretes 6 principal hormones. You can remember them as flat PG. Now let us discuss them one by one, starting with follicle stimulating hormone. This hormone promotes the development of ovarian follicles and consequent production of estrogen in females, whereas in males it stimulates spermatogenesis. Next is luteinizing hormone. A surge of LH in females in cooperation with FSH induces ovulation at mid-cycle. It also maintains the corpus luteum after ovulation and secretes progesterone, also known as pregnancy hormone. In males, it stimulates the interstitial Leydig cells in the testis to produce testosterone. The third one is adrenocorticotropic hormone, ACTH. ACTH, also known as corticotropin, is a single chain peptide hormone that consists of around 39 amino acids and is derived from a larger precursor molecule. It stimulates the adrenal cortex to secrete glucocorticoids, mainly cortisol, and also small amounts of sex hormones like androgens and estrogen. Next is the thyroid stimulating hormone. The TSH is also known as thyrotropin. It is a glycoprotein hormone that consists of 2 polypeptide chains, alpha and beta, with alpha chain being identical to that found in LH, FSH and human chorionic gonadotropin. Its primary action is to order the thyroid gland to secrete the thyroid hormones like triiodothyronine and thyroxine, that's T4. It also is involved in stimulation of thyroid iodide uptake, increased synthesis of thyroidal storage protein, that's thyroglobulin, and stimulation of T3 and T4. This is achieved by an increase in the thickness of the follicular epithelium and vascularity of the thyroid gland. Talking about the prolactin, prolactin is a single chain peptide made up of around 198 amino acids with a chemical structure very similar to that of the growth hormone. It is principally involved in cooperation with other hormones for the development of female breasts and in the initiation and maintenance of lactation, that is milk production, shortly after childbirth. It is released from the pituitary mammotroph cells in increasing amounts during pregnancy under the influence of circulating estrogen and acutely in response to suckling by means of sensory neuroreflex arising in the nipples. Now, let us talk about the broad spectrum growth hormone. Growth hormone, also known as somatotropin, is the most abundant of the pituitary tropic hormones, but unlike others, it can influence a variety of target tissues. It is a single chain peptide containing 191 amino acids with 2 disulfide bridges. Growth hormone promotes linear length of bones, particularly longer ones, by stimulating the growth and calcification of epiphyseal cartilage cells. It also influences the growth of visceral organs, adipose and connective tissues, endocrine glands, and striated muscles. Growth hormone levels are at their highest during early childhood and particularly at puberty, where it is essential for normal gonadal development, and then its levels steadily decline throughout the life, thereby implying that growth hormone may not be essential for normal human adult health. Now, let us talk about the secretions of the posterior lobe. Remember, the posterior lobe does not synthesize hormones, but it contains a large number of unmyelinated nerve fibers. These fibers form the hypothalamic hypophyseal tract within the pituitary stalk. These neurons synthesize predominantly 2 non-peptide hormones that are 9 amino acids long, vasopressin and oxytocin. These 2 hormones are then transported as protein-bound secretory granules, that is neurohypophysin, down the nerve fiber to accumulate at the nerve terminals within the posterior lobe. The hormones are then released by exocytosis into the capillary bloodstream, running close to the nerve terminal swellings. Now, let us have a look on vasopressin. Vasopressin is also called as arginine vasopressin, that is AVP. The principal action of vasopressin is to stimulate the reabsorption of water from the distal convoluted tubules and collecting ducts of the kidney, thereby reducing urine volume and conserving the body fluids. Vasopressin, therefore, also referred to as antidiuretic hormone. This effect is mediated by specific V-type AVP receptors on the tubular cells surface linked to the rise in the intracellular CMP, that is the second messenger. Now, let us conclude this lecture by talking about the next hormone stored in the posterior lobe, that is oxytocin. Oxytocin has a similar structure to vasopressin, but it has distinctly different physiological functions and second messenger system. Although it is present in both sexes, but its effects are well understood only in females. Oxytocin stimulates the rhythmic contractions of the uterine smooth muscles during childbirth. It also stimulates milk ejection from the lactating breast in response to the infant suckling by contraction of the myoepithelial cells present in the alveolar secretory epithelium. Oxytocin also contributes but is not essential for the initiation or maintenance of labor mediated via specific oxytocin receptor on target cells. Dear students, this is all about today's lecture. Hope you have understood well. Thank you and goodbye.